don't have any sound for Amadou. Okay, so I can go ahead. Thank you. So hello everybody. We would like to welcome you to this session that I am going to share with my brother Amadou Gaye. He's we are from the same country, Senegal, but he's living abroad in the US. And it is a great pleasure for me to meet him virtually and to co-share this session. I would like to congratulate all the people that are behind this wonderful organization of this meeting, particularly the Tanzanian Society of Human Genetics, the African Society of Human Genetics, and the H3 Africa Consortium. So in this session, we will be having two invited speakers, Zane Lombard, that I'm going to introduce. So Zane Lombard obtained her PhD in human genetics in 2018 with her research focusing on bioinformatics-driven approach to disease gene discovery. She's currently a principal medical scientist and the team lead of the research and development group in the division of human genetics, where the focus is on the implementation of next generation technology for genetic diagnosis. So Dr. Zane Lombard, we are very pleased to introduce you. You have the floor for your presentation.
Hello, everyone, and thank you for joining this session. I want to thank the organizers of this meeting for the invitation to present to you today. Details on this research project called DDD Africa, where we are exploring the utility of diagnostic exome sequencing in an African setting. I'm Zane Lombard, the PI of the project, but the work I will present today is entirely a collaborative team effort from the whole DDD Africa team listed below. Developmental disorders represent a spectrum of lifelong disorders that are often presented from childhood and that impact an adaptive function on all three domains. This is conceptual, like language skills, social, as well as practical function. Developmental disorders include a spectrum of impairments, of which intellectual disability is the most common. It's thought that approximately half of developmental disorders are due to a genetic cause, and in severe cases, up to 80% of disorders could be genetic. However, genetic diagnosis is not straightforward, as this set of disorders are marked by both clinical and genetic heterogeneity. This Circos plot gives you a glimpse of this complexity uncovered by the DDD UK study, showcasing the variety of different clinical features oftentimes seen in developmental disorders, and the variety of genetic loci found to be linked to these. We know that despite several decades of research and development, about half of children with a developmental disorder remain without a diagnosis. And unfortunately, the picture in Africa is much more dire. In South Africa, the majority of the country does not have access to genetic services, and we fall far short from the global recommendation for the number of medical genetic specialists and genetic counselors required to offer an appropriate service. Based on an internal patient file review that one of our students performed, focused on our genetics clinics, we see that the majority of patients who visit our clinics have a developmental disorder, and of those, 92% remain undiagnosed. So why is this important to get a diagnosis? Well, we've seen clear evidence that early and accurate diagnoses can be life-changing for patients with developmental disorders. Identifying a genetic etiology informs parents and other family members of potential future risks and improves clinical management. And even in situations where a diagnosis does not represent a cure, which is often the case, the end of the diagnostic odyssey has significant psychosocial impact for families. Families of children with undiagnosed genetic diseases often face an uncertain and unpredictable journey that can last for several years and with significant financial cost. This is a global issue, but even more pertinent in low resource settings. Here you can see a graph representing patients from our genetics clinic and the number of years as well as the money spent on this diagnostic odyssey. For many of our patients, the cost of cascade testing quickly moves beyond the cost of an exome sequence test, which could potentially yield results much faster. I realize that I don't have to convince delegates of this meeting of the revolutionary impact next generation sequencing and specifically exome sequencing has had on the field of genomic medicine. But given the scarcity of resources on the African continent, we're left with the question of whether exome sequencing is a practical first-line diagnostic strategy for developmental disorders in low and middle-income countries. This is then the main focus of the DDD Africa project, where our rationale is to map the genetic etiology of developmental disorders in Africans, whilst catalyzing improved diagnostic services. This is a collaborative study across 2 African sites in Johannesburg, South Africa, as well as in Kinshasa in the Democratic Republic of Congo. And there's a strong partnership with the DDD UK group, and they have already demonstrated the value of adopting a comprehensive genome-wide strategy to elucidate the underlying causes of developmental disorders. DDD Africa forms part of the Africa Consortium, and alongside our goals of performing whole exome sequencing on 500 patient-parent trios, we are also focusing on training, capacity building, as well as community engagement. One of these activities that everyone has found extremely informative are our monthly clinical review and variant interpretation meetings. Originally, we planned to have these take place in person, but we've mostly had to move this to a virtual platform due to the COVID pandemic. This has had the positive effect that at least more students have had the opportunity to participate, and the discussions and learning opportunities have been plenty. So in the DDD Africa project, we've established definitive inclusion criteria to ensure that we are enrolling patients from the more severe part of the spectrum of developmental disorders, as these patients are more likely to have a genetic cause for their disorders. These include patients with moderate to severe intellectual disability, as well as those with a specific major or minor organ anomaly. Patients excluded include those with a suspected multifactorial or environmental origin, and known conditions with no strong evidence for a monogenic cause. We're capturing all our clinical data on the Decipher database. This is an online tool which has ensured secure data sharing between sites and also implements fair data principles. Our patients represent the full spectrum of ethno-linguistic languages that are spoken across South Africa and the DRC. What you can see here in the inner part of the pie chart are the array of languages spoken by participants from the South African site, with 90% of participants being of African ancestry. For the DRC side, Lingala is the most common language and ethnic group represented. Our study also implements a tiered informed consenting process, giving participants control over decision-making regarding the reuse of data and samples, as well as the use of medical photographs captured as part of the clinical workup process. We believe that our approach is working well, and this is really reflected in the different decisions being taken, as shown here on the right. For instance, what you can see in the bottom pie chart is that the majority of patients are agreeing to broad sharing of data and samples, but are making much more reserved decisions and a much more variety of decisions when it comes to the use of photographs of their underage children. Regarding the sequencing and bioinformatics arm of the project, we've really been leveraging the strength of the DDD UK team whilst ensuring that we build appropriate capacity and expertise locally. We've had excellent support from the H3A BioNet's local network in setting up our bioinformatics pipelines. Thus far, we've sequenced 480 participants using the Wellcome Sanger Institute's Illumina NovaSeq platform. In addition, we've also done a small pilot project here in South Africa to establish the requirements for performing whole exome sequencing on our locally available INS5 instrument. So following a preliminary quality control step, all data that is generated in the UK are transferred to the WITS high performance cluster and are then further shared with the DRC team. We have set up an automated bioinformatics pipeline locally aligned to international standard practices, which is followed by then a manual clinical interpretation process. The pipeline has been implemented in Nextflow and documented on GitHub. Over the next few slides, I will present some of the preliminary findings from our first analysis of our whole exome sequencing data set. I just want you to note that I've also included some patient photographs in the coming slides where consent was given, of course, as the medical geneticists and dysmorphology experts in the audience may appreciate the opportunity to compare and contrast key phenotypic features with some of their own patients. So overall, we obtained excellent quality data with a median depth of 55x. The number of mean and novel variants that we observe in our exome sequencing data set are actually significantly higher compared to exomes from the European patients shown on the left here. And this can be expected given our knowledge of the genomic diversity of African individuals and due to the fact that we are using the standard Build 38 genome reference and not a more sophisticated pan-genome or genome graph at this stage. The downstream implications of this is that our filtering process will result in many more variants to consider than is the case with other patients. As you can see here on the right, at the bottom of the pyramid, we're left with a manageable number of variants previously implicated in developmental disorders for the majority of the exomes. These are our so-called low-hanging fruits, and that would be investigated first. However, if none of these variants turn out to be likely disease-causing, we have a much more involved investigative task to complete. The following results that I'm going to present will focus on the analysis that we've performed using our small nucleotide variant and indel pipeline. We plan to also evaluate de novo mutations, copy number variants, as well as cases of suspected mosaicism and UPD. And several of our trainees involved in DDD Africa are working on implementing these additional analyses pipelines in the meantime. So from our in-house whole exome sequencing study, we were able to identify putative disease-causing mutations in 6 of the 7 patients, and we were able to validate these variants in 5 of the patients. This investigation identified a patient with Takanushi-Kozaki syndrome, which to our knowledge is the first reported case in Africa. This is patient 1. We also identified 2 cases of mandibulofacial dysostosis with microcephaly in patients 3 and 7, followed by congenital muscular dystrophy and mental retardation type B2 in patients 4 and 5 who are fraternal twins carrying the same compound heterozygote mutation. Patient 1 was initially referred to the genetics clinic at the Rahima Musa Mother and Child Hospital in Johannesburg, South Africa, at the age of 6 months for assessment, and this was on the basis of her cardiac disease and associated dysmorphic features. A cytoscan HD array was performed and reported as negative, and several other investigations did not yield any feasible answers. She was enrolled in DDD Africa at 10 years of age. The identified de novo mutation in the CDC42 gene identified using exome sequencing was classified as likely pathogenic based on its absence in population databases, the fact that there were 2 pathogenic submissions in ClinVar, as well as in silico functional study evidence showing the variant to have deleterious effect. Based on the diagnosis made, we were able to make further recommendations for care and end this long diagnostic odyssey for this particular patient. The second case I want to share with you demonstrates the scope of African genomic studies to improve clinical dysmorphology. This is a 9-year-old female patient of North African descent with moderate intellectual disability, ventricular septal defects, hypertonia, dysmorphic features, macrocephaly, as well as short stature. Our clinical team posited a diagnosis of Cornelia de Lange syndrome based on these phenotypic features, but our exome analysis actually revealed an ARID1B frameshift deletion, with evidence suggesting it to be likely pathogenic. So we know that mutations in ARID1B are known to cause Coffin-Cyrus syndrome, which is a rare clinical heterogeneous disorder. Our clinical team noted that this patient actually presents with very specific features that may present a distinct African gestalt, which could be utilized to update and refine dysmorphology analysis. The suggestion was to follow up with additional phenotyping before we confirm this diagnosis. We also noted that with Coffin-Cyrus syndrome and other related syndromes, that they present with distinctive DNA methylation episignatures that have demonstrated diagnostic utility. And we are currently looking into opportunities to confirm if this is also applicable to our patients, and this information can then be used in future to resolve ambiguous clinical cases or to reclassify those with variants of unknown significance, and even to identify previously undiagnosed subjects through targeted population screening. The final case shows the utility of exome sequencing also to confirm clinical diagnoses. This 6-year-old patient was born with a limb defect and syndactyly, and both mother and child have intellectual disability, with mom also presenting with a tongue abnormality and cleft lip, brachydactyly, as well as renal cysts. Our clinical team suggested that the clinical presentation aligned with oral facial digital syndrome type 1, and this diagnosis was actually confirmed by the exome sequencing. And of course, that presented a service that we were not able to offer previously to these patients. We're hoping to confirm a diagnosis in the mother in future, as we know that adults with idiopathic intellectual disability oftentimes have not had genetic testing done, as is also the case here. So I hope that today that I've demonstrated to you that our preliminary findings underscore the potential of an exome sequencing diagnostic approach for rare developmental disorders in a resource-constrained environment. We're gathering evidence that show that whole exome sequencing will be cost-effective and beneficial to patient care in the long term. And although this test is currently still costed higher than traditional testing options, the question really is not how much will it cost, but rather how much will we save in the long term, both for the patient and the family, and also in the local public healthcare system. Over and above this, proving feasibility of local implementation would allow us to step into the future and towards genomic medicine services in Africa, and also lead to job creation and local skills development. I would really like to thank the NIH and as well as some of our local funding bodies for providing funding for this study. I'm also very thankful to our many excellent collaborators, both in the AfSHG Africa Consortium and beyond. And a special thank you also to the DDD Africa Research and Administrative team. Just want to end off by saying that if you're interested in learning more about the DDD Africa study, or even to possibly become a partner site, please feel free to email me, or you can reach out to me on any of the social media channels provided in my bio on the online conferencing system. Thank you so much for your attention, and I'm happy to answer any questions that you have.
Thank you very much. Thank you very much, Dr. Gane. It was a very nice and interesting talk about development disorders. I think that it it is a huge challenge in Africa, particularly in West Africa, where molecular diagnoses are not available. So I think that the DDD Africa project will be very interesting tools for all countries in Africa to work together in order to allow these patients to be diagnosed. So thank you very much for this interesting talk. Maybe we will wait at the end of the different talks to have a Q&A session. So the next presenter will be introduced by Dr. Amadou Gaye.
Yeah, hi, good morning, everyone. Very happy to be here with you and to be chairing this session. Thank you to Rokhaya for the initial introduction. Without losing more time, we would like to introduce Professor Leon Mutesa. Professor Leon Mutesa obtained his medical degree from the University of Rwanda in 2003, and he obtained a PhD from the University of Liège, which is in Belgium, in 2009. And after his studies, he did serve as a director of the Department of Clinical Laboratory Department at the Kigali University Teaching Hospital, and that is the largest referral hospital in Rwanda. He has been also a director of the Medical Research Center in Rwanda, Biomedical Center, which is part of the Ministry of Health. We have to say that Professor Mutesa actually pioneered new developments and implementation of a reference center for medical genetics in Rwanda. And currently this is the only one that is existing in the East African region. He is currently the director of the Center for Human Genetics of the College of Medicine and Health Sciences at the University of Rwanda. Leon has really been a member of has been member of a lot of expert committees, and he has led various regional and international research consortia. So he's a very experienced member of the African scientific community. Please join me to welcome Professor Leon Mutesa. Hopefully you enjoy his talk and gain from his knowledge. Thank you very much. Leon, the mic is yours.
Yeah, thank you very much for this opportunity to share this talk today. So I'm going to just give you an overview regarding the genetic diagnostic of neurodevelopmental diseases, some challenges and perspectives. So the developmental disorders, also known as a DD, they are disorders that are characterized by severe and chronic disabilities attributable to mental and or physical impairments in childhood. Most of the DD they have impact on the adaptive function in all three major domains, and these domains include the conceptual skills in the language and the reasoning and memory. There are also those affecting the social interpersonal skills, and others the practical functioning of self-management and the personal care. When you look at the causes, the genetic causes of DD are predicted to be present in 25 to 50% of diagnosed cases. Moreover, more than 50% of children with severe DD of probable genetic origin remain without a genetic diagnosis using classic techniques like karyotype, other chromosomal microarray. And even the recent study have shown that we need just to use the more advanced techniques just to be able just to identify the majority of DD with de novo mutations or new genes, yes, causing these disorders. Well, when you look at the prevalence, we see that most of the DD, they are characterized by slower learning, and the attention deficit hyperactivity disorders more common in these disorders. Language disorder and the speech with speech delay, speech impairment. It's also most of the clinical characteristics of the DD. And even more patients, they present a global development delay, intellectual disability. Autism spectrum disorder is also found in most of the DD, affecting the childhood. And permanent sensorineural deafness is one of also the signs that we find in these disorders. Well, there are two groups that we see that these disorders are being classified. So there's a group of externalizing disorders, which is characterized by outward directed behavior, where we found the non-compliance, aggressiveness, hyperactivity, impulsiveness symptoms. And this includes especially the ADHD, conduct disorder, oppositional defiant disorders, and among others. And most common disorder we have here is the Fragile X syndrome, which is the most common intellectual disability disorder in boys. And then there's a group of internalizing disorders where we see the disorder being characterized by inward focused behavior, where patients, they turn to express this depression, anxiety, social withdrawal. And it includes childhood anxiety and mood disorders. And the more common disorder here is the Rett syndrome, which is commonly found in girls. When we use the DSM-5 childhood disorder tool, we have different characteristics, different groups. There's a first group, which is a group of intellectual disability. In this group, we found the Fragile X syndrome, which is most common inherited form of intellectual disability, and with a high prevalence in boys and girls, where you see the patient that are being characterized by dysmorphic features, large head, endocrinocephaly, long face, chin, forehead, chin, ears, and behavior problems characterized by hyperactivities. And we also have in this group the most common chromosome abnormality, which is Down syndrome, most common in Africa as well, where the patient, they have of course the intellectual disability and a variety of dysmorphic features that are being characterized in childhood, especially by psychomotor development delay. In this group, there are also other syndromes that we see with autosomal recessive condition or X-linked condition. And then there's another group of specific learning disorders, which is characterized by neurodevelopmental disorder that begins by school age. All facts might not be recognized until later. However, 5 to 15% of school-age children struggle with learning disabilities. So we see there's a delay, school just learning disability, and a delay characterizing these children at this age. And then there's a combined group of autism spectrum and ADHD disorders, where we see the global learning difficulties, specific learning difficulties characterized by dyslexia, dyscalculia, dyspraxia, which is characterized by coordination difficulties of movement, speech delay disorders, and tic disorders. And then there's another group of motor disorders. These disorders are characterized by the disorders of that cause abnormal and involuntary movement, like the ataxia syndromes, cervical dystonia, chorea, Parkinsonism, Rett syndrome. Rett syndrome in girls characterized by involuntary movements. This syndrome, the more progressive neurodevelopment disorders, it can be diagnosed very late. So it's during the development of the child, so the symptom cannot really appear before 6 to 18 months. And then later, you see there's a developmental stagnation and the regression of the development, and then the child starts just to have these motor disorders with involuntary movement of the fingers, the hands, as you can see here on the picture. And then we have the feeding and the eating disorders. In this group, we see the patient having symptoms characterized by persistent disturbance of eating and eating-related behavior that results in alteration of consumption or absorption of food, which lead to rumination disorder, anorexia nervosa, bulimia nervosa, and the most common disorder is the Prader-Willi syndrome. And this disorder is characterized by severe hypotonia, feeding difficulties in early childhood. And later, they develop excessive eating and gradual development of morbid obesity. And they also express the speech delay, speech impairment, cognitive impairment, and behavior problems characterized by aggressivity and other behavior problems. So more common disorder as well. And of course, there are other two groups characterized by disruptive and impulse control and the conduct disorder, and also a group of anxiety disorders, which is more mental disorder, characterized also by hallucination or depression. Well, these DDD group disorders, they present a diagnostic challenge. When you look at on a genetic background, so there's a trend to be influenced, to have an influence on DD, and which is related to these challenges. The chromosome and the gene disorders, for example, the Fragile X anomaly occurs in 2 to 3% of children with autism spectrum disorder. And when you look at this autism spectrum, they have 5% of elevated risk for chromosome abnormalities. And about 25% of children with, for example, the tuberous sclerosis disorder, they have autism spectrum disorders. And when you look at them in the diagnosis, only 50% of DD can be diagnosed using the classic routine techniques like a karyotype, Sanger sequencing, CGH, and others. And then there's a big proportion of disorder, DD disorder, that we are not really diagnosed using this classic routine. So that's how we need a more advanced techniques like NGS to be able just to diagnose these disorders. Well, the recent study, UK study, which is DDD study, Dicephalic Developmental Disorder, have been able just to detect the genetic causes of the DD disorders by using the whole exome sequencing. So they screened more than 4,000 DD families, which were most of these patients were characterized by intellectual disability, the DDD, epilepsy, autism spectrum disorder. And then they have been able just to identify de novo mutation, and 30% to 40% of cases have been just confirmed. So this just showed that different regions that were just detected as a new, and they could just identify new candidate genes and mutations which were not characterized in, when they were using the classic techniques. And this just showed that how we can just diagnose the DD by NGS and also be able just to identify new loci or new genes. So here you can see a number of CNV or even genes that have been related to development delay, growth delay, and also speech impairment. This was very contributive study, which allowed us to identify new genes that are related to DD disorders. In our setting in Rwanda, when we started just to provide the medical genetic services almost now 15 years ago, so we have just conducted a study now 7 years ago on identification of DD genetic causing by using the RCGH analysis in a patient who were presenting development delay, intellectual disability with multiple congenital anomalies. So several chromosomal rearrangement, duplication, deletions have been identified in this group. And then we remained, of course, with a group of patients where we could not just identify any genetic causes using these techniques. And then we later selected this group to use, to make analysis of genomic analysis of the unidentified variants. So you can see here there are three families of interest that we studied here. The first two families are consanguineous families, and the third one, all of those, the third one is not consanguineous families, but all of those families, they had just in common some characteristics, clinical characteristics characterized by communication disorders, autism spectrum, learning disorders, motor disorders, ADHD, or intellectual disability. So the aim of this study was just to identify the pathogenic variant responsible for neurodevelopment disorders in these families using whole exome sequencing. So, of course, as I mentioned, the prior analysis were negative. The chromosomal microarray was negative. And then these are our patients. You can see here, they were similar to previously published patients where some mutations have been identified. So, but for this group, we could not just identify the same mutation like the similar patient here published previously. And then we identified a mutation in this group of patients. So there's a VPS 51 biallelic variant, which was just causing the microcephaly with brain malformation in most of these patients, especially the two families, consanguineous families, where the patient turns just to have similar symptoms. So this was just the first time to identify these variants in such type of patients. So in conclusion, so establishing a genetic diagnosis of D for the development disorders has numerous implications. beyond just medical treatment, and they should be considered as a priority for patient care and for discovery of novel genetic causes and the mechanism, like the de novo mutation or inherited mutation. And of course, the African founder mutations also can be found. Non-causative mutation related to DD, also another interest, a benefit, and the frequently mutated genes in the clinical subtypes of DD. Given the diagnostic rate of genetic testing and the potential for clinical and personal utility, there's a consensus that genetic testing should be offered to all patients with global development delay, intellectual disability, and or autism spectrum disorder. And of course, this needs just to create a network and a collaborative with collaborative groups for addressing the issue of accessing the next generation sequencing techniques, which are not available for most of the African countries. And this can just be a solution for how we can diagnose the DD in Africa. Well, I want just to thank most of my collaborators from Europe, the Center for Human Genetics in Liège, and who are just supporting the initiative for DD studies in Rwanda. Professor Kati and Kati Anastos and Phil Casso from Albert Einstein College of Medicine in New York, who are just contributing for the biobank, and others like Professor Johnson Muschlegel and Janet Watke, who are supporting the diagnostic testing, and of course, my colleagues from Center for Human Genetics. Thank you very much for your kind attention.
Thank you very much, Amadou.
Sorry, I was having a problem with my microphone. And thank you very much, Leon. And we'll proceed to the next speaker. Our next speaker is Aissata. Aissata Touré from Mali. She is a research fellow based at the Neurogenetics Lab at the University of Techniques and Technologies of Bamako. She's currently a resident in neurology at the National Teaching Hospital, which is located at Point G. She has also an interest on hereditary neurological diseases. And Aissata is also a member of the neurogenetics domain and is currently working on the clinical and genetic aspect of spinal muscular atrophy in a Malian population. So please welcome Aissata Touré. She's going to talk now. Thank you very much.
Hello everyone, my name is Aissata Touré, student at the University of Sciences, Techniques and Technology of Bamako. It is a real pleasure and a great honor for me during this 13th conference of the African Society of Human Genetics about genetics of spinal muscular atrophy in the Malian population. As outlined, we will go through the background in which we will have a little detail on spinal muscular atrophy, then the objectives of this work follows. Spinal muscular atrophy is an autosomal recessive disorder characterized by the degeneration of the motor neuron in the spinal cord. It's due to the mutation of the SMN1 gene. The homozygous deletion of the SMN1 gene accounts for 98% of cases. It is the most common cause of autosomal recessive inherited mortality. Its frequency is lower in Africans compared to other populations, with an asymptomatic carrier frequency of 1 per 30 to 1 per 50 in European population and 1 per 209 in Mali. The clinical features include symmetrical progressive muscle atrophy and weakness, predominantly proximal, absent reflexes, more accentuated in the lower limbs, tongue fasciculation, hand tremor at rest, lower limbs, and spinal deformities. There are 5 subtypes from 0 to 4 classified according to the age of onset and the maximum motor function affected. There is not a lot of data on spinal muscular atrophy in Africa, precisely in sub-Saharan Africa. We report here genetically confirmed cases of spinal muscular atrophy in the Malian population. Our objectives are to clinically characterize families with spinal muscular atrophy phenotype and identify the genetic defect. Our study, approved by the Ethics Committee of Faculty of Medicine and Dentistry of Bamako, written consent obtained from the participants before their enrollment after a long explanation of the study course. Patients carefully evaluated by neurologists and other practitioners such as pediatricians, geneticists, and other specialists if needed. Socio-demographic data and clinical features were collected and mentioned in medical record. Blood tests including creatine kinase were performed to exclude other causes. DNA was extracted from the peripheral blood by using standard procedures. The quantitative PCR method was performed for dosage of SMN1 and SMN2 gene, which analyzes and examines exon 7 of each SMN1 and SMN2 gene, as well as dosage control gene. As a result, we had 5 families totaling 8 patients with spinal muscular atrophy features and consanguineous marriage in 3 families. Genetics analyses in 3 index patients show with homozygous deletion of SMN1 in all. 2 and 3 copies of SMN2 has been found in patients with type 2 SMA and more than 4 copies in those with type 3 SMA. The observation 1 concerns 24-year twin brother from consanguineous marriage, pointing out the recessive inheritance pattern. They was seen for weakness in forelimbs started at 12 years old with fatigue when walking and difficulty climbing stairs. Clinical examination found more proximal weakness in forelimbs, absent reflexes, and no sensory loss. As shown by this picture, we can note a uppers and lowers amyotrophy, also tendon retraction in lower limbs. Genetic testing identified the homozygous deletion of the SMN1 gene with more than 4 copies of SMN2 gene, evoking SMA type 3. The patient went under physiotherapy with slow worsening evolution. Here in observation 2, we are 4 and 2 years old boys, referred for suspicion of muscular disease. Pregnancy and delivery were normal. There is no consanguinity, but both parents are of the same ethnic group. Symptoms started at 6 months with hypotonia and later with difficulties sitting with falls. He could not switch from lying to sitting himself. The pedigree showing 2 affected males and 1 suspected female from a non-consanguineous marriage with an affected parents, assuming a recessive inheritance pattern of the condition. Clinical examination found a normal cognition, axial hypotonia, scoliosis when they are sitting, generalized amyotrophy predominant in lower limbs, weakness with fasciculation, and absent reflexes. Genetics testing identified homozygous deletion of the SMN1 gene with 3 copies of SMN2 gene. The patient index died at 10 years old due to pulmonary infection. Our last observation concerned 3-year-old girl from consanguineous marriage. She was seen for walking difficulty, started at age of 4 months with hypotonia. She could not hold the head. This pedigree showing the affected patient from unaffected parents in consanguineous marriage, pointing out a recessive inheritance pattern. Clinical examination found a normal cognition, generalized hypotonia, accentuated dorsal kyphosis and scoliosis, absent reflexes. And then genetic testing identified homozygous deletion of the SMN1 gene with 2 copies of the SMN2 gene. This is the first such study reporting that high number of symptomatic cases of spinal muscular atrophy in Mali. Despite the low carrier frequency, our study shows that this disorder is not uncommon in Mali. Through the high frequency of type 1 described elsewhere, no case has been found in our study. This may be due, in one hand, to the low size of our cohort, and in the other hand, the delay of diagnosis. Patients die before being seen by a doctor. In conclusion, extensive genetic testing of putative cases may uncover many more cases, which could advocate for treatment availability in our country. Future genotypes-phenotype studies may identify other genetic modifiers. We cannot finish without thanking our partners, our participating families, and all the research team of Mali, especially thanks to Dr. Landouri.
Thank you very much, Dr. Aissatou Touré, for this excellent presentation. So now we're going to move to the next one, presented by Andrea Regina Sens, and I'm pleased to introduce her. She's one of my students. She's one of the rising stars in human genetics. And we have a lot of hope about her in our team. So she's actually, she has been, she has her diploma of technology in applied biology and a bachelor in bioengineering and biotechnology at Cheikh Anta Diop University. And she's actually doing a master's degree in human genetics in our faculty. She's planning to be PhD next year.
Hello, everyone. I would like to thank the African Society of Human Genetics. It's a great honor to have the opportunity to present our work. My name is Andrea Regina Nilansen, and here is a report of the activities of the Division of Human Genetics of Dakar on identifying chromosomal abnormalities in patients with suspected congenital disease in Senegal. Genetic disorders are generally fall into 3 main domains: the single gene defects, the multifactorial conditions, and the chromosomal abnormalities. These chromosomal abnormalities are defined as a structural or numerical alteration in one or more chromosomes. They are common. In effect, 0.4 to 0.9% of newborns have chromosomal abnormalities, and half of them have an abnormal phenotype. The chromosomal abnormalities generally fall into two main categories: the structural abnormalities and the numerical abnormalities. The structural abnormalities are genomic rearrangements on one or more chromosomes. They result from an unequal exchange between chromosomes or misrepair of true chromosome breakage, leading to different types of abnormalities like translocation. We have, for example, the translocation between the chromosomes 9 and 22 in the acute myeloid leukemia with fusion genes. We also have deletions, for example, 5p- syndrome, duplication, and more. The numerical abnormalities are most common than the structural and consist in a gain or a loss of one or more chromosomes. They are divided into two types, the aneuploidy and the polyploidy. The aneuploidy is the presence of an abnormal number of chromosomes. For examples, we have the Down syndrome with 3 chromosome 21 and the Turner syndrome with a less of one of the X chromosome. The polyploidy is a condition of possessing more than 2 complete sets of chromosomes. For examples, we have triploidy with 69 chromosomes instead of 46, and tetraploidy with 92 chromosomes. The chromosomal abnormalities can affect both autosomes and gonosomes and may occur either as a result of error in the germline cells with mutation in the first or the second divisions, called non-disjunction of the chromosomes, or may happen after, during the embryogenesis, in the mitosis, in which case only a proportion of cells will be affected. We talk about mosaicism. The normal sexual development is a synergic orchestration of numerous. It implies 2 distinct processes, the sex determination and the sex differentiation. The sex determination takes place at the genetic phase with the fertilization. At this moment, we have the XX or the XY given by the parents and the differentiated gonads. Because the XX or the XY are similar gonads, they need differentiation in male or female organs. That is the sex differentiation with the influence of genes like AfSHG. Any problem happen in the normal sexual development can lead to disorders of sex development, DSD. There are congenital conditions defined as discordance in the chromosomal, gonadal, and anatomical sex. They affect 1 on 2,000 to 1 on 4,500 children born each year. They are the result of a deviation from the typical progression of sex development, including genetic variation, atypical development programming, and change in hormones. The SRY gene, sex-determining region, localized on the chromosome Y, the short arm of the chromosome Y, triggers the cascade of molecular events leading to male sex development. In effect, the SRY gene functions as a transcription factor, testing determining factor, TDF, leading to the differentiation of the B potential gonads into testes during the 6th week of gestation. This image shows the influence of the SRY gene on the indifferent gonads. In the presence of the SRY gene, the indifferent gonad differentiates into testes for the male sex development. When the SRY gene is absent, The indifferent gonad differentiate into ovary for the female sex development. The diagnosis of the chromosomal abnormalities. Many new molecular techniques have been developed for chromosome analysis, but the karyotyping is still the gold standard for chromosome analysis. It allows to identify the numerical chromosome anomalies and some structural abnormalities, and it also gives the genetic sex. The molecular screening is based on the polymerase chain reaction, PCR, and it permits the search of specific genes. In our case, the SRY gene. In Senegal, the socioeconomic and the cultural aspects impact the management of these conditions. We are in a poor resource country, and the diagnosis of congenital disease were expensive because samples of patients were sent abroad for chromosomal analysis. In a society when the first question new parents and family asking is, it is a boy or a girl, the question remains unanswered for newborn with sexual ambiguity. Their condition become a taboo subject while they need the diagnostic of their condition and the confirmation of the genetic sex. Based on this observation, since the 9th meeting of the African Society of Human Genetics in 2016 in Dakar, efforts were made for having an infrastructure for patient care. And then the Human Genetic Division was created in 2018. Our aim is to do the report of the cytogenetic diagnostic activities of the newly implemented Division of Human Genetics of the Faculty of Medicine, Pharmacy, and Dentistry at the Cheikh Anta Diop University of Dakar. The experimental approach was a case studies. From January 2020 to May 2021, patients were referred to the Division of Human Genetics from different hospitals in Senegal. They were referred for karyotyping and/or molecular screening of the SRY gene. The clinical evaluation is the first step in the diagnosis. It is based on the physical signs and symptoms that lead to the suspicion of chromosomal abnormalities or the observation of sexual ambiguity. The procedure of the standard karyotyping is the following. We take first 5 milliliters of venous blood of the patient. This total blood is placed in a culture medium, the lymphomedium, for a culture at 37 degrees for 3 days. After these 3 days, we use colchicine, a mitosis blocker that arrests cells in metaphase, and we proceed with hypotonic shock to break the nuclear membranes. Then, the cells are fixed with a mixture of methanol and acetic acid, spread onto slides by dropping, digest with trypsin and stay with GEMSA, and we analyze the mitosis with the SmartType software for having the karyotype. The screening of the SRY gene is based on the PCR, with the use of specific primers and a specific PCR program. The results we obtain are the following. For a total of 30 patients, we had cases, 80 cases of numerical abnormalities, and 7 cases of disorders of sex development. The age group of our study population were as shown in this table. For 0 to 2 years, we have 40 cases. 2 years to 12, 4 cases. 12, 5 cases. And for the 18s and others, 7. The chromosomal abnormalities observed in our patient are listed in this table. For the autosomal abnormalities, we have 8 cases of Down syndrome, 2 cases of Edwards syndrome, and 1 case of monosomy 18 in mosaic. For the gonosomal abnormalities, we have 6 cases of Turner syndrome. For the 6th case, we have 5 cases in mosaic, and we have 1 case of Klinefelter syndromes. Here we have the karyotype we done in the lab with the SmartType software. So here we have an example of patients with Down syndrome. On the right, we have a female with the Down syndrome, and on the left, a male with Down syndrome. This is a karyotype of a male with Edwards syndrome. We can see the 318 chromosome. This is a female with mosaic. We have 2 population of cells, 80% of 46XX and 90% of 40 minus 18. This is a type of patient with Turner syndrome in mosaic. We have 80% of 46XX and 20% of 45X. And then we have a karyotype of a patient with Klinefelter syndrome. For the screening of the SRY gene, we have here the result of an electrophoretic migration on agarose gene of PCR products for the SRY gene screening. So for the patient P1 and P4, the SRY gene is present. We see a band at 400 base pair, and for the patient P2 and P3, the SRY gene is absent. The test is negative, confirmed by the negative control. For the 9 patients with disorder of sex development, the result of the search of the SRY gene were confirmed by the karyotype. All the patients with a SRY gene positive had a karyotype 46XY, and all the patients with SRY gene negative had a karyotype 46XX, except for 1 patient that we're going to talk about later on. Concerning the age group of our study population, the majority of our patients had between 0 to 2 years. Their conditions were diagnosed at birth because we don't have a prenatal diagnosis in Senegal. While in certain countries like South Africa, European or American country, the prenatal diagnosis is done for the chromosomal abnormalities. The second group, age group of 8 years and older, represents 23.3% of our population. This shows that there is a late diagnosis of chromosomal abnormalities and also DSD because the poor information the population have about patient care. A further reason lies is the fact that the DSD are a taboo subject in our society. Our results show also a high frequency of chromosomal abnormalities. The most frequent we have in our population is the Down syndrome, with 44.4%. Of course, this is the most common autosomal trisomy, and it affects 1 on 700 live births. The second most frequent was the Turner syndrome, with 23.3%. This is the most common sex chromosomal abnormality in females and the most common genetic cause of primary amenorrhea. In our population, we had 5 cases in mosaic, and these mosaics were very considerably from individual to individual, from mild to severe signs and symptoms. We also had 2 cases of Edwards syndrome. This is the second most autosomal trisomy, and it affects 1 or 3,000 to 1 or 50,000 live births. We had also one case of Klinefelter syndrome. It affects 1 of 500 to 1 on 1,000 newborn males, but it's still underdiagnosed in the African context. In our population, we have found a monosomy 18 in mosaic. This is a deletion of one of the chromosome 18, and our patient had symptoms of an homogene Edwards syndrome. In the literature, we only found a monosomy 18p, and it affects 1 on 50,000 live-born infants with a very moderate and non-specific symptoms. We also found in our population a very interesting case. In fact, it's a patient with a karyotype 46XY, 46XX that we have considered as a chimera. The patient was referred to the lab because of sexual ambiguity. So we done the karyotype, and then the screening of the SRY gene came out negative. The chimera is the fusion of two different zygotes in a single embryo. But for our patient, we found that her mother have the same karyotype. So the question is, can we still talk about chimera? More investigation are needed, such as genotyping. The determination of the genetic sex in patients with DSD is crucial in our cultural and religious context. In fact, the parents give the name of the baby 7 days after the birth. The karyotype and the molecular screening of the SRY gene permit to obtain the genetic sex in few days. In addition to the SRY gene on the sex chromosome, on the Y chromosome, other genes localized on autosomes have been implicated in the sexual development. Genes like WT1 localized on the chromosome 11, SF1 localized on the chromosome 9, SOX9 on the chromosome 17, and other genes. These genes may be needed to be screened. So in conclusion, we can say that our work highlights the fact that cytogenetic diagnosis and molecular screening is vital for patients with suspected congenital disease in Senegal. The management of these patients needs to be individualized, particularly for decisions related to sex rearing, surgical intervention, hormonal treatment, and potential for fertility preservation. Multidisciplinary health care is also essential for a better diagnosis. Also, the only one structure specialized in genetic disease, the Division of Human Genetics, need to be localized in the different region, but also needs equipment and new technologies upgrades like CGRH or FISH. I would like to close my talk with acknowledgements to the African Society of Human Genetics, the Faculty of Medicine, Pharmacy, and Dentistry of Cheikh Anta Diop University, as well the Senegalese Society of Human Genetics. Thanks for your attention.
Thank you very much, Andrea, for this very nice talk, highlighting the capacities that is under building in Senegal for diagnosis for these developmental disorders. So we have ended the presentation that we have in this session. Very interesting presentation, talking about a subject that is a challenge in Africa, as we said before. It is important to localize in sub-Saharan Africa to be equipped for molecular testing. I think that through the presentations that have been done, we know what is available in Mali, Senegal, and Rwanda, but also what is underdoing in South Africa through the DD Africa project. And I think that if we put all together our strength, we will be able to build up capacities for molecular diagnosis in Africa. So it was a real pleasure to be in this session, and we will be having the Q&A session. So I have some questions, but I will come later with. Maybe Amadou, if you want to speak, you have the floor.
The opportunities and some of the challenges they faced by most of those researchers working on from the African soil. At this point, I think I have only one question for Professor Lawan Mutesa, and I wanted to know if there is a way, I assume before referring people for genetic tests, what ways do you use to distinguish developmental disorders from behaviors that might be stemming from psychosocial issues related to the child's environment? Because you mentioned ADHD and some other external clues that you guys have. How do you distinguish the ones that might be genetic to the ones that are really could be due to some psychosocial issues?
Thank you, Amadou. We have some attendees that would like to speak. The first one is Alassane Di Bane Maiga. So Alassane, you have the floor for your question.
Oh, do we need the presenters to answer the questions before we move to the next question, or do we compile all the questions and then let them answer?
You're right. Yeah, we can do that. Yeah.
I'm not sure I heard just
I think Professor Dane Lombard is online. Andrea.
Oh, okay. Lewan has left. That's not a problem. Okay, if you have questions for Professor Zane Lombard, then your questions are welcome. It seems like the other presenters are not anymore in the room.
Okay, I don't have a question for Professor Zane Lombard, maybe a suggestion. I would like to thank her for giving us this very nice talk and highlighting this DD Africa project. I think that, as we said, many countries have challenges regarding genetic diagnosis for DD, and it will be good that we join this DD Africa project, trying to build the capacity in the different countries. But even if in those countries capacities are not yet available, I think that through collaboration in the different cases that will be diagnosed, that will be suspected with DD, we can try to find a way to share samples for diagnosis. So Professor Lombard, what do you think about this? We are really open to this kind of collaboration because every day in our we have patients suspected for DD, and for those for whom we don't have capacities to do the diagnosis. And what we do is to send samples to providers, sequencing providers, to have it. And most of the time, we have to pay for this by ourselves for research purpose. So if it will be possible to have this kind of collaboration, to share samples for diagnosis, it will be of great interest for us.
Thank you. Thank you, Rakaya. Yes, I agree with you. I think working in collaboration with each other and building networks for this kind of diagnostic service is really important. And I think it is possible. I think already we're connecting with one another in the community through the African Society, through other local human genetic societies, and through AfSHG. So I think the DDD Africa project is building resources for how we can, in a financially viable way, collect the right kind of clinical information, collect the DNA samples, do DNA extractions, etc. And I think as the network builds, we also build the collective buying power to negotiate with sequence facilities and with sequence instrument suppliers to make sure that we can have access to this kind of technology on the continent and at different centers across Africa. So yes, I think the important thing is to build the network and to work together, and hopefully through some of these initiatives, we're already getting there. One of the things that we've been developing as part of DDD Africa and as AfSHG is the African Rare Diseases Initiative, to try and band together and build resources specifically for rare genetic disorders, and to work together to do those things like you outlined. So I'm happy, again, if anybody wants to email me or contact me, or work through the AfSHG Consortium to connect and start building this network and capacity even more so across Africa. That'd be great.
Professor Zane, so we will keep in touch for this collaboration. Any question? We have one question in the Q&A live that was raised by Professor Julie McKinney. So she was asking about spinal muscular atrophy. So through the presentation from Aissatou Touré. So she said, please comment on gene therapy products approved. Are you participating in gene therapy trials? We already have an answer from Gida Londure, but maybe if Gida would like to add something through this discussion. Gida, you have the floor if you want to add something. No, so maybe he's not logged in. Okay. So another question is from Professor Michel Ramsey. Thank you very much for all the presentation. Having a balance between testing specific mutation and then NGS is important. But mutation testing is more cost effective. If there is a common mutation, how do we get more molecular data across African population to understand the mutation? Very interesting. Maybe Professor Zane, you can answer or give some inputs about.
Thanks, Rakaya. Yes, I can maybe just add a few things. It's a very good point that Michelle makes. We do need more African data, and I think the only way to do that is to build these resources, research resources and cohorts that we can get more exome and whole genome sequence data from, actually, to really understand. I mean, I do take the point that at the moment NGS is still not very cost effective or available to many people. One of the ways that we're dealing with it in our department is that we've opted for a gene panel approach, where we have gathered several different genes that are involved with inherited diseases onto a panel, and we run that on all of our patients, and then do selective bioinformatics analysis depending on the patient's characteristics as a cost-saving approach. So, and that also then helps us to build more molecular data that we can use in our databases to interpret findings. So I think there are ways to gather this. I think, again, working together and pooling resources and pooling data together is much more useful than it being stuck in little pockets across the continent. So that's one thing that we could look at, is to pool specific NGS data and other mutation data that we have together to really make interpretation a bit easier.
Thanks, Professor Zane. Just to add a few comments, I think that it is, as Professor Ramsey says, it is important to do testing, mutation testing, specific mutation testing. It is important to do it in the African context because it is low cost effective. Maybe for the panel of genes that are actually used, most of the mutation that have been reported have been done in other populations that may be not specific to African population. So through the different efforts that are under construction in the different countries, maybe through the pan-genome, when the genetic diversity of African population will be much more dissected, we may identify specific mutation for each of the DD, and then it will be much more easier to do genetic, to do diagnosis for those populations. So I think that as everybody said, it is important that we work together to have it done. Yeah, thank you. I see that Professor Gida is logged on. So Professor Gida, if you have some comments.
Yes, thank you very much. Yeah, you can see my mosquito nets. I am in the village. So you can also hear children around. I say hi to everyone, faces we haven't seen for a long time. So to answer this comment or question, I don't know, in Africa, SMA has now treatment since 2017. Two treatments recently came out, and more are coming out. But unfortunately, we do not available, at least in Mali. So it is expensive and out of reach, even not covered by any insurance company here. So we have reached out to NGOs or other other organizations working on treatment equity. So we are to a way to get treatment for those patients, because it is nonsense to in research, and when the treatment is to not have a way, at least give some medicine to under population. So yeah, I work on that. Regarding Michelle's comments, Yeah, it is true. Zane has said, and Okaya also has said that there is a need to kind of develop something for Africa, or not develop, but another way of testing African. If I refer to the genetic analysis we have done here locally, if we take gene panels to do our testing, we have less than 40% of success rate. So meaning that for about 6% of our families do not have the mutation described in already established gene panels. So I even went in one of the spastic paraplegia panel. We don't see, for example, a gene that was identified in one single Malian family and another family in Brazil. So meaning that those panels do not work for Africa. So the way is to go directly. Now we are not doing panels because they are expensive. Like we pay $2,000 for one sample, and we come out. It comes back with no results. So we just go for exome sequencing. But for exome sequencing also, as Zane said, it's true per sample. It's more accessible for the cost of genotyping, but the analysis also is a challenge. Is a challenge because filters, again, the filters that are available, the reference genome that is available might not work for African samples. So Emi might be here. He might comment, and Zane also might comment that there are disease working group is working with Genomad to kind of find a way to insert African variants in the international database so that the filtering can be more efficient for African samples, because many millions of SNPs that were found in the African samples are not in the reference databases. So the answer is to do more and more exome sequencing. I know Michelle will say to share. This all is based on really the limited resources we all have. The African investigators, myself, are really let's say I'm uneasy to go wide in. We have our analysis capacity is so limited that or our early, people might be able to analyze faster than us, and what we could publish earlier. Then I think it's a matter of finding a way to help African researchers to speed up these capacities. So this is all about again, infrastructure, knowledge sharing. Yeah.
Thank you, Gida. You're absolutely right. I think that we in Africa, most of our challenge actually for genetic diagnosis is the data analysis. And we have to build the capacity for this in each country. And we have a question from Emil Lumaka about data sharing. So he said that data sharing is already problematic and for non-clinical data. More difficult for clinical data. How would you approach this in Africa? Amadou, do you want to add some comments?
Well, I think it will be very unfair for me to comment on that because I'm not sure if the realities that we faced here in terms of data sharing are the same in Africa. It would be maybe more appropriate for someone there to tell us what are the challenges in data sharing in this area. And then maybe the person who asked the question can tell us why he thinks data sharing might be even more problematic for this type of data we're talking about.
Thank you, Amadou. If Dr. Emelumaka is online and want to give more insight into his question. Dr. Emel? Okay. Okay. Maybe before he's in the session, I have a question for Gida. Yeah. Maybe not for Gida directly, but for Aissata Toure. So she talked about spinal muscular atrophy. And just last week, we had a patient where it was suspected spastic paraplegia, and it seemed that it is the same pathology because the same gene or the same genes are involved, SMN1 and SMN2. So one of the questions is, Gida, do you have in Mali facilities for testing, for genetic testing of the deletion for spastic paraplegia or for the same pathology? And the other question is that the same disease? Is a spinal muscular atrophy the same disease as spastic paraplegia? Because I'm not a clinician and I would like to know more about this. Gila.
Okay, I don't know if Aissata is online to comment on the last question. Okay, so first, spinal muscular atrophy is different from spastic paraplegia. Spinal muscular atrophy is a motor neuron disease, a lower motor neuron disease, whereas spastic paraplegia is an upper motor neuron disease. So it's true that sometimes at the early stage, people might confuse those diseases, but if you examine closely your patient, when you can see that while SMA is a kind of floppy baby, SPG comes like a rigidity, spasticity. So, but it can be that an SMA patient of already prenatal weakness, delivery can be complicated and cause brain damage, what we call infirmité motrice cerebral palsy. So cerebral palsy can come with SMA, but these are really completely different diseases. So do we have a way to test them here? yes, we have a way to test them in our lab, but we did not start doing it because we still have a student at NIH helping with the molecular testing that comes a little bit cheaper for us to send DNA than using our own reagents here. But we have the quantitative RT-PCR machine. We have all the reagents that are needed to do it.
Thank you very much, Gida. Professor Zane, do you have some comments?
So I can maybe comment on Ame's question about data sharing. I understand his point. I think we all have difficulties with data sharing because of maybe restrictions in-country restrictions about what kind of data can be shared and with who. But I also think about the point that Gita made, that it's sometimes really difficult for the group who was very instrumental in collecting data to also use that resource before it is shared to a larger community. And that is also important. I think it is something that we need to take into consideration when we think about data sharing and doing research together, is that there needs to be some level of agreement and equity amongst the groups that are sharing the data to make sure that the resources are used for the purpose that it was firstly collected, and to make sure that the local communities where the data is collected from will receive some kind of benefit from it before it is shared outside of those groups. So it is difficult to do that within and across Africa just because of the differences in access and infrastructure. But I think there are great, especially great bioinformatics infrastructure being built and skill sets and capacity development happening. And I think some of the tools that BioNet is working on that implements these FAIR principles, and like the Decipher database that we're using in DDD Africa, it facilitates it and makes it easier to share data within collaborative groups, and to make sure that the data is collected in the correct way and shared in the way that it was supposed to be. Bioinformatics infrastructure and bioinformatics tools, I think, play a really important role in that.
Thank you, Professor Zane. I see that Dr. Emelu Maka is online, but he has some issues with his microphone. Eme, if you...
To piggyback,
that's okay now? Okay.
is he on?
Dr. Eme, you have the floor for your comments. Okay.
I mean, based on what Zani said, it comes to my mind an initiative that is at the NHLBI that I was involved in called TopMed, which I never thought that can happen in this country, where each PI thinks they have enough resources and can deal with their data, and sometimes they're more reluctant to sharing. In Africa, the context is very different, as Kira said. Everyone can borrow a little bit of strength from other people. And from what Zane said, maybe that TopMed idea can be kind of adapted to the African context. Because if the issue is, if I release my data before I publish, other people who have more resources can analyze the data and publish before me, then all these PIs within even the African Society of Human Genetics, they're already starting to have the infrastructure through the BioNet. What can they do then is agree together to put their data together, but then each PI or each researcher would have a say on what questions would be examined. Of course, that is already dealt with in the consent, but then prioritizing the questions and the analysis. For example, if you bring your data from Mali as GIDA into that pool of African scientists, then you would have some kind of priority on what questions you want to investigate first. And then the crunching of the data, the analysis, you can get help from other people, but then you are the prime candidate to investigate your questions first before someone else. That means other can investigate your data, but not with the questions that you decided that you're interested in first. That way, the data is shared, but each of you guys have a say and can decide to investigate certain questions that they are more interested in before other people. That could be, I think, one solution to do it.
Thanks.
I think this is really, an approach that might work. Yeah, as I always tell people when I share my data, or I say we are working on very rare families, and if they are going to take each single variant they will find in the families we study and put them outside, it will demonize the work we are doing locally. Maybe, you know, maybe to kind of do the collaboration of the research question for the original investigator and see how to answer that question, and then do the work for the general question. I think this approach can really work.
Hello.
Hello, Emmy.
Yeah, thank you to the technical team. They have been able to bring me on the main stage. And Amadou, I think what we are saying here, thank you for asking me to comment. It's a discussion that is being going on since a few years now, that we are discussing how to get access to the data, how to generate this research that would allow us to improve the interpretation of data for rare diseases in individual of African descent. Most of the time we approach this question as researchers, and then we see the MTAs, and then we see how the risks are for our research, not always for the patient, the risk for patients, because when you aggregate data, there's a way to minimize those risks. But there is another approach that we don't often take into account, which is the patient and the family aspect. We are conducting those researches on the continent. I myself, I'm not only a medical doctor and a researcher, I'm also a father of a child with a problem. And my child has a genetic diagnosis, and I know what it takes to reach a genetic diagnosis. And oftentimes, when we are receiving requests from researchers that are working out of Africa, they have identified a new mutation in African patients. They are willing to know the frequency so that they can be able to achieve the diagnosis or not. I feel that the need is there. I feel the stress of those parents. It's something that, as a researchers, from that point of view, we don't know what it takes for a parent to have a child to be waiting for 5-10 years without having a diagnosis, even when the whole exome sequencing or the whole genome sequencing data are available. So I think there is a need to try to, for parents and community of people that are directly affected by rare diseases, to come and sit with researchers, try to present that different perspective. I think it's another way of explaining the problem to people, because it does it beyond our research interest. It's really a matter of health for families and the community. There's a way we can help with data we have, and I think people will be much more flexible if they are in contact with parents, with families that are affected by those diseases. It's my own case. I have this problem in my family, and I also work closely with the family. I see them in eyes, and I discuss with them. If I have data, I would not hold my data for whatever reason, except if it's unethical. I think we need to change the prospect of the discussion, bring the community, the people that are affected, to bring them on the table to discuss. It will change a lot of things. This is just above what Gida and Zan have mentioned.
Thank you very much, Eme. Yes, I think that maybe through the H3 Africa Bionet and through the different working group, for example, for the rare disease working group, through collaboration around the different countries and for those patients for which we have data available, what we can do is to pull them together for data sharing, but maybe securely stored in order to implement data analysis, to build the capacity on data analysis in our different countries. Maybe in that way it will be much more easier, because as Emma said, having access to data is very difficult in our context. But if we work together, generate data, secure this data, and then building capacity around analysis, we will be able to have our own reference sequence, maybe much more specific to African countries, and we will be able to do diagnosis for these very rare disease, taking into account the genetic diversity of African population.
Please, Rukaya, I'm not on the panel, just to add something quickly. In all fairness, it's not that we are in the nowhere. There's already something that has been developed, a discussion that we have with the H3 Bionet, as Zani mentioned. Now with those discussions, we are at a level where we are designing how this would work, how this beacon for rare diseases would work. Now it's about calling people to share those data, so that we can be able to fill this research that we are developing with the H3 Bionet. We need data to populate this research, not only data from the reference population, but the near future, we also need data from patients. I need to know that a particular mutation associated with disease has been identified in a patient with developmental disorders in South Africa, so that when I observe the same mutation in a Congolese patient, I quickly reach a diagnosis. I don't need to go through the whole process. I just filter the known mutation and then identify this. This patient will be solved in one day instead of waiting years. So we need those resources. Please, this is a call, a plea. We need the resources so that we can fill the resource.
Yes, thank you, Eme. I think that all of us involved in this work, I think, are willing to release data if they exist. Yeah, I know that in Senegal we don't have yet, but I think in the future we are actually building capacity for next generation sequencing. So it will be maybe possible to be involved in this data sharing. And Gida too, I think maybe. So it left for us only 3 minutes for this session. If you would like somebody who would like to give few comments before we close, Amadou.
Not really, no comments. Just want to thank all of you people who attended today for the lively discussion. And I'm hoping also that in the next meeting, we will have a whole session dedicating only to this issue. Because as Gita and Dumeka said, it has been years now we've talking about this data sharing. It has been very theoretical. It might be good in the next African Society of Human Genetics sessions, conferences, to just block a whole session where we discuss about the feasibility of a solution and then discuss it more concretely and see how this data can be pulled together without each of the investigators losing out somehow. And Lumeka said, at the center of this is also the patients. We need to do something. Okay, as scientists, people want to publish, we have to preserve that need, but we also think about the patients, and we can only achieve something and come up with a solution and start moving the ball, rolling the ball, if we put it at the center at one of the conference and discuss it, and everyone comes up, what are their concerns? How can we address your concerns so that you would agree to putting your data into this pool, and knowing that you gain something and the rest would also gain, because you cannot mine the whole data. You can only investigate the questions that are priority for you, and the data is useful for other people to investigate questions that are also relevant for patients, for them. That's all the comments I had on this data sharing issue, and hopefully we can discuss it and start rolling the ball at some point.
Thanks, Amadou. I think that the next organizers of the African Society of Human Genetics meeting will take into account your propositions. So we will be having more time to discuss about this very important topic. So thanks very much to all the presenters that gave us a very nice talk. Thanks for all the participants that have given their comments. It was a real pleasure for us to be with you in this session, and hope to see you in person soon. Thanks everybody, and have a nice meeting.
Thank you.
Thank you. Bye.
Bye-bye.